Hello, good day, everyone. So again, I am here, Teacher Denso, and I will teach you some information regarding to the biology. So, but before that, so this day is our asynchronous day. So it's time for our asynchronous day. It's time for our discussion. It's time for gaining some information regarding biology. So today, I'm going to discuss all about the genetic materials. So first, what are the things behind the genetic materials? First, we have the genes. The gene or genes. So when we say genes, no, genes is an important segment of the deoxyribonucleic acid or the DNA that signify a unique protein that controls a specific function in the cell. So a gene is capable of storing genetic information and sometimes can undergo mutation. So this is the example of the picture of the gene. So we locate the gene on the chromosomes. Human DNA consists of about 3 billion bases, and more than 99% of those bases are the same in all people. For higher forms of organism, the DNA is the genetic material that contains all the genetic information. So, DNA is responsible in controlling all the cellular activities within living organism. So, DNA can be found in mitochondria, or what we call the mitochondria DNA, or the mtDNA. Okay? And uh, also, we can locate the DNA, or the mostly no, in nucleus, or what we call the nuclear DNA. There are four DNA bases. So what are the four bases? That is the adenine, cytosine, guanine, and thymine. So adenine or adenine and guanine are classified as urine since they are double helix ring structural, while the cytosine and thymine are classified pyrimidines or pyrimidines, all right, which contain the centering structure. So we have the nucleotides in the building blocks of the DNA. It is made up of four bases, a five carbon sugar, the deoxyribose, and the phosphate group. Okay? The group or sequence of these bases determine the information available for building and maintaining an organism, similar to the way in which letter of the alphabet appear in a certain order to form words and sentences. Each base is also attached to the sugar molecules and a phosphate molecules. Together, base, sugar, and phosphate are called nucleotides. Okay? This nitrogenous or nitrogenous base, a complementary base pairing A to T and G to C, or adenine to thymine and guanine to cytosine, which pair up to form a new strand of DNA following the semi-conservative process of replication. So, in 1953, okay, James and Watson, James Watson and Francis Crick developed the structure of a DNA as the double helical structure, since the structure contains two strands widening around each other. The double helix DNA is the winding staircase. The structure contains two sugar phosphate, the DNA base pair that are like or linked by hydrogen bands. Okay? So, this is the picture of the DNA, the double helical structure. And we have here the nitrogenous bases, which is the adenine, guanine, thymine, and cytosine, which stands for different color. So, let's talk about the DNA replication. Okay, just see the picture. So how does the DNA make a copy of itself? The process starts when the two strands of DNA separate from one another when enzymes break the hydrogen bonds between the complementary nitrogen bases. Then DNA double helix is unwound by an enzyme called the helicase. Again, helicase, the DNA double helix is unwound by an enzymes that the enzymes what we call the helicase. This separation forms the replication fork that takes a Y-shaped configuration, as you can see on the picture. Okay, the helicase, the arrowhead, no, which making or break down or unwound the strands. Okay? Then the RNA primer binds to the leading strand run in 3 to 5 direction. Okay? This RNA primer act as the starting point of synthesis. DNA DNA polymerase binds to this strand and directs the addition of new complementary bases of the TAGC of the bases. Pairing produce complementary nucleotide, showing the presence of one parent strand and a new daughter strand. This form of replication is considered to be semi-conservative, okay? Since it contains one original strand and a new daughter molecule strand, several RNA primer binds to different points of the lagging strand. The direction of synthesis still runs from 5 to 3 direction, thus making several discontinuous segment or what we call the Okazaki fragment. DNA legates joins these newly formed discontinuous fragments. So, alamin natin or let us know regarding RNA. So, when we say RNA, the ribonucleic acid, or commonly known as the RNA, contains a long strand made up of nucleotides compared to DNA. RNA has a single chain and does not form a double helix structure. The comparison between RNA and DNA you will see on this picture. Okay? So, the four bases are the guanine, cytosine, adenine, and uracil. So, at this time, at this point, thymine is not present. So, it replaced by the uracil. Instead, thymine it replaced by the uracil. Okay, so let's see the comparison of the two. So the comparison of the RNA and DNA, okay, when it comes to sugar, bases, strands, and helix. First, on sugar, DNA have the deoxyribose. RNA only has a ribose, okay? Deo, from the word deo, double, parang dalawa, right? And sa basis na, kung on the DNA presents the thymine, RNA replaced the thymine replaced by the uracil. Okay, for strands, DNA is double strand and RNA is single strand. In helix, DNA is a helical structure and RNA is not or no. Let us proceed to the protein synthesis. So, in protein synthesis, as you can see there, protein found inside the cell are responsible in controlling activities of the cell. In what way do DNA and RNA work together to make a unique protein that will perform a specific function? Protein synthesis starts when the messenger RNA or the mRNA transcribes and copies the genetic information in making protein from the DNA strands. This process occurs inside the nucleus. The mRNA strand exits the nucleus via the nuclear membrane. Then the ribosomal RNA or the rRNA, the copied information from the mRNA as codon code, which contain three consecutive nitrogen bases. Okay. Next one, we have the translation. So, translation take place when the transfer RNA or the tRNA start to bring anticodon or complementary copy of the mRNA codon to the ribosomes. Each anticodon contains a single type of amino acids. Amino acids are known as the single blocks of protein. Translation stops when all codons are converted into amino acid and when a specific protein molecule is produced. So that is all about the protein synthesis. As you can see on the picture, I put there one, starting from 1 to 8, the process of the protein, or that is the general process of the protein synthesis. Okay, so that's it for now. God bless you. Uh, I think that you gain a lot of information again coming from me. So continue study and study well. Focus on what you are doing and what you are studying. So again, I am Teacher Renza, and God bless you, and thank you for your watching.